they're so happy to be here. All right, so take a minute and set up your page. This is Unit 10, Biochemistry. And what is today? Monday. 3/8/21. Only one more day of masks, maybe. What period is this? 7. Okay, so we're doing B9A, and this is the last part for B9A. So it says you're going to compare the functions of different types of biomolecules. Write it clearly. Again, I'll be taking this for a grade on your next test. So this will probably be about 20 points on your next test. Compare the functions of different types of biomolecules including we're going to hit nucleic acids today. Okay, so I'm going to backtrack just a minute. 4 biomolecules, 4 major biomolecules: nucleic acids, proteins, lipids, carbohydrates. So carbohydrates, lipids, proteins, nucleic acids. Yes? Those are the big 4. There are more biological molecules inside of different kinds and stuff, but these are the big 4 categories that fit in there. You might want to grab a couple of colors to do this. I'm going to do this in a couple of different colors just to make sure that I can do this. If you have your colors, if you don't have colors, there's some over there in the container over there that'll help. Sometimes even if it's just the little squares and dots and stuff that you put in color, that'll help you kind of organize things. So we start off with a square. If you need to move closer, go ahead and move closer. Shift your desk around so you can see more easily. So here, we're going to do nucleic acids. Okay, so this, whenever we talk about nucleic acids, it's all about the genetic material. We're going to start it today, and then in the next unit, what we're going to do is we're going to talk about how is the DNA replicated, and how is the DNA, exactly what's the process to making a protein. So this is going to be, this stuff that we're doing now is, there's a lot of it, but that's not incredibly complex. The next unit we're going to do is a lot more complex than this. How many different types of nucleic acids are there? Can you think of what they are? What are the 2 nucleic acids? There are 2 of them, DNA and RNA, so there are only 2 nucleic acids on the planet. And they are, what did you say they were? Okay, I'm going to put RNA first, and I'm going to put DNA right under it for a purpose. Okay, so RNA equals, and I'm going to skip a little space over here, ribonucleic acid. Okay, and I'm going to pop this up a little bit. Ribonucleic acid, right? DNA, we put deoxy, and then we put ribonucleic acid. So if I cover up the deoxy, they're the same thing, right? But deoxy means take away what? Oxygen. We take the ribose, here's the ribose. If it ends in OSE, it's a sugar, right? So we take away, we take the ribose sugar that's a part of it, and we take away an oxygen, and it makes it into deoxyribose sugar. And so that's what's going on there. And that's why it does a little bit of weird stuff here. Now, for each of these, again, we're going to do the structure and the function. So structure. I'm going to have to shrink this back down a little bit. So again, every one of these we did, here's the big square, we did the structure, we did the function, we did examples. Structure, function, examples. Structure, function, examples. Okay, so we're hitting the same thing over and over again on these guys. So the structure is going to be pretty complex here. It's made of sugar, phosphate, and a nitrogenous base. Do you guys know what a base is? Do you know what the pH scale is? Yeah, pH scale talks about how acidic it is, right? So an acid has a pH of like 0 to 7, right in that area. A base has a pH of 7 to 14. So acids and bases are chemically opposites of each other. We talk about them that way. So this nitrogenous base is actually a little bit basic here, but overall we call them nucleic acids. Now, they have the basic, you know how I keep asking you what's the basic part of, what are proteins made of? They're made of amino acids, right? So the monomer of a protein is an amino acid. It's a chunk of it, right? And you get a whole bunch of amino acids hooked together and you get a protein. Here, in this case, the monomer of DNA, how many is mono? Is called a nucleotide. And it's made of, I'm going to put a box around that in a different color. That nucleotide monomer is made of one nitrogenous base Okay, so the nucleotide is made of one nitrogenous base, one phosphate, and one sugar. So that's what the nucleotide is made of. Okay, did you guys get that? So let me show you, you guys have all seen this before, over and over and over again. Double helix, right? Twisted ladder. So look at the sides here. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, so sugar, phosphate, and then the little black thing right here, there is the nitrogenous base. So this is one unit of it. Here on the other side, sugar, phosphate, nitrogenous base, there's a unit. Okay? Here's the next unit, right here. Next unit down. Sugar, sugar, phosphate, nitrogenous base. Yeah? Notice that the white ones are always hooked up to the black ones. That orange thing is just a connector. The white is always hooked up to the black, and this greenish color is always hooked up to the gray. That matters. Okay? So this is what it's talking about when it does the DNA. So the monomer, okay, DNA is a polymer, right? Poly means there are lots of parts to it, and they're repeating parts. That's what a polymer is. So here's the one basic part. You get a whole bunch of nucleotides together, and that forms a DNA. So if I say what is DNA made of, you're going to say, oh, they're made of nucleotides, right? What are proteins made of? Amino acids. So amino acids are the monomer of that. Okay, now what we're going to do is we're going to look closely, and we're going to compare DNA and RNA. All right, so we have the deoxyribose sugar. That's the name of the sugar. We have a phosphate group. And we have a nitrogenous base. A nitrogenous base, nitrogenous, what element does it have in it? Yeah. So I'm going to put a little asterisk, a little star beside this one, and I want you to realize that there are 4 nitrogenous bases in DNA, but there's an extra one that's in RNA. So here we have A, T, C, G, and I want you to darken those. Those are not element symbols, obviously, right? These are actually abbreviations. So the real name for this is adenine, thymine, cytosine, and guanine. These are the ones you have to memorize, A, T, C, and G. Now, here's the kicker with this. A's and T's always hook up together. C's and G's always hook up together. If you accidentally hook up an A to a C, there's your mutation. You just made a mutation in there. That's a problem. That's one way to make a mutation there. So A's and T's always hook up together. C's and G's always hook up together. So what do I mean by that? Well, let's go right here, and I'm going to get on the line here, and I'm going to put sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Let me go one more. Phosphate, sugar. There. Makes me happy. So I did 1, 2, 3, 4, 5 sugars and 4 phosphates. So sugar, phosphate, sugar, phosphate. That's the way it goes down the line. And I'm going to do the exact same thing. I'm going to give myself some space here, and I'm going to do the exact same thing on the opposite side. They need to match up with each other, line up with each other. Okay, so there is a covalent bond between the sugar and, let's say I'm going to put an A there first, adenine. Phosphate doesn't bond to anything but sugars. The next sugar, I'm going to say, hey, let's put a C there. That's going to bond to that. And I'm going to just put some letters. That would be part of my DNA strand right there. That's on one side. On the other side, again, I have a covalent bond. Now, which one of these hook up with adenine all the time? Which one hooks up with A? T, so I'm going to put a T here. Which one hooks up with C? G, so there's always going to be a G opposite of that. Which one hooks up with C? And T? Yep. You see how that is? And G? There you go. Now, I'm going to put one more piece of information here. So in order to hook the halves of the ladder together, they're not true chemical bonds. Remember talking about covalent and ionic bonds? Yes, no. Yes? Okay, these are not real chemical bonds. So instead of putting a line, which is what we usually indicate for that, I'm going to put 3 little dots here. Between C and G, they have 2 because I need to make it right. So either 2 or 3 little dots. You put a couple of little dots through there and we'll be okay. Now I'm going to grab another color and I'm going to talk about those little dots for a minute. Okay, so does this yellow show up? No? Okay. Okay, let's take the green then. There we go. Okay, so these little dots are not chemical bonds. They're not covalent bonds. They're not ionic bonds. The coolest thing ever, these are actually called hydrogen bonds. And you guys have first-hand experience with this. They're intermolecular forces. So what I mean by that is, here's a molecule, here's a separate molecule, and there's a force of attraction between them. Usually it's like a little electric charge between them. And I say you have experience with this because water has this. Have you ever seen bugs where they're like walking on top of the water? There's some really cool pictures. If you get close-up pictures of them, their feet are not wet. They're not in the water. You can see the water bend in where their feet are there. So if I were to take a bug off the surface of the water like that and put him on a paper towel, there would be no drips of water at all. It wouldn't be wet at all. Because the bug isn't inside the water, it's actually walking on the surface, and the surface is almost like it's got this skin on it, and it bends in where the bug is walking on it. And the reason it does that is because this hydrogen molecule, the hydrogen on one water molecule, is attracted to the oxygen on another molecule. There's this electrostatic charge, this attraction between them. It's really mild, real minor, but because of that, bugs can walk on the surface of the water. Because of that, did you guys ever in grade school, did you take like a paper clip or a pen and set it on the surface of the water? Did anybody do that? You'll probably do that next year in creative chemistry. But it takes a little careful practice, but it's easy to do. And it's not that the paper clip, you obviously know if I just drop a paper clip in water, it's going to sink in the water, right? It's heavier, it's more dense than the water. But if you're really gentle and careful and you don't break the surface tension of the water, things called hydrogen bonds. The reason this is so important, this is so hugely important in both chemistry and biology, is because they're not truly chemically bonded here, we don't have to set fire to it, we don't have to throw chemicals on it to pull these apart. All you have to do is put a little bit of energy on there and you can bump them apart. Now, on the one hand, that means maybe you could have some mutations there, but on the other hand, this means that, hey, when I have this DNA strand, I don't need to go through a chemical reaction. I can just gently bump these apart, and I can spread this ladder into two. So when we go through mitosis, remember how we had to make a copy of all the chromosomes, right? This is a chromosome. When you go through mitosis, it splits this in half. One half builds its own other half of the ladder. The other half builds its own half of the ladder, and so now you have two strands of the DNA, and that's how it works. We're going to talk about that again in the next unit, but that's basically what's going on. And the only reason it can do that is because it's not chemically bonded to each other here, it's just held gently together by a hydrogen bond through there. And when I say gently, it's gently, but it means it doesn't take a lot to break, but how many of them are there? Well, if your DNA is 10,000 things long, 10,000 A's, G's, C's, and T's long, that's a lot of hydrogen bonds holding those things together. So it gets to be pretty well tightly held together, right? Okay, so these little things are the hydrogen bonds that are in here. All right, now let's go over here and work with the RNA. Instead of deoxyribose sugar, cut off the deoxy, and RNA has ribose sugar. It still has the phosphate, and it also has the nitrogenous base. Okay, but because we messed with the oxygens, this one has one more oxygen in its sugar than this one, it's not going to be able to bond with things the exact same way. So here, the number one difference that we have here is that the nitrogenous base in RNA, U, takes the place of T, so uracil. So this is in RNA. So every time there should be a T, pop it off and put in a U, uracil. So 1, it has U instead of T. 2, it has one more oxygen than the deoxyribose, right? Deoxy means take away an oxygen. And 3, it's only half the ladder. So instead of having two sides to the ladder, you have one half of it. So let's build one here. So let's say sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So this one's going to be, and let's say I build this off of this one, okay? So if I made a copy of this, just one side of it, what bonds to A? Well, if it's in DNA, T bonds to it, right? But if it's in RNA, what would bond to this? U. Here's my next one. What bonds to C? G. Next one says C. What bonds to that? G. Fourth one says T. What bonds to T? A. And the last one says G. What bonds to G? C. So you just made a copy of one side of it, didn't you? It's like you built the other half of it, right? But every place a T should have been, you put a U. That's how RNA is different from the DNA. 3 things. 1, RNA has one more oxygen than the sugar. 2, it has uracil instead of thymine because it can't bond to the thymine. And 3, it has only one half the ladder. And that's a great thing because when we start talking about how we make proteins, you have to have one side open to do that. Okay, the next thing, and you're going to see this on the test, is they're going to ask you to fill in the other half of the DNA strand. Or they'll ask you to fill in that, like if they give you part of a DNA strand, they'll say, hey, what would the other RNA strand be? So pay attention because they're going to ask you to fill it in like it's DNA or match it up like it's RNA. So one of the two. So let's do DNA. So let's say I have A, C, C, G, T, T, A. What bonds to A? This is going to be DNA, so T, and then what?
G.
Uh-huh. You got it? Yeah, fill in the rest of that. It's pretty simple. You just remember A's and T's and C's and G's. The curved letters hook up together. The A and the T, it's at. So it hooks up together too. So A's and T's go together, C's and G's go together. Now, there's one more piece of information I'm going to throw at you. You don't have to memorize it. The only reason I'm showing you this is because you're going to see it on the test paper, and I don't want it to throw you off. They're going to add 5' to 3' end, and then on the bottom it'll be 3', just the opposite to 5'. So you're going to see this, it looks like 5 inches. It's called 5' and 3', yeah? And they might even say in the question, moving from the 3' end to the 5' end. Yeah, you don't care. You don't have to know that. Just don't be freaked out when you see it. It's just a part of it. They're being accurate, but you don't study that until you get into college. 5', 3' end, you just don't care. But it's on there, okay? Now, last little thing on here, we've got the structure. Let's do the function and the examples. So the function, there you go. The function, twofold. It stores and transmits genetic info. I mean, you're going to use it to make new cells, to make new sex cells, body cells, whatever. But to be honest, if I were going to say this, I think it's more important for me in my brain to say that this is what has the instructions to make all your proteins. Every protein that's in the body, the instructions for that are here in the DNA. Every single protein. And then the process that we use to make the protein, the DNA has the instructions, the RNA is the one that's actually used to make the instructions. Because the DNA has to stay safe inside the nucleus. It's got to be kept safe in there. You don't want it mutated, right? So we keep it safe inside the nucleus. The RNA is the one that runs in, makes a copy really quick, takes it back on the cytoplasm, and that's how the proteins are made. Okay? The last thing on here, examples. You already know these, though. What are the 2 examples? Yeah. This is just to keep it consistent. Okay, once you have that written down, flip to page 11 in your packet. Page 11, we're looking at the right-hand side, the nucleic acids exit.